We're going to take a look at translation. We're going to use these messenger RNA codons when we do translation, and that's going to code for the different amino acids, so we know which ones to put in which order to make the protein. And in all of the organisms that we've studied, all of them use the same genetic code, so the genetic code is universal. We have one start codon. That's the only one that you need to memorize. That's AUG, and it codes for the amino acid methionine. We do have 3 stop codons, which you don't have to memorize, and then we have these other codons that will code for the 20 different amino acids. The genetic code is what we call degenerate, which just means that it's redundant. So if you look at this bottom right-hand corner, you can see that we've actually got 4 different messenger RNA codons, but they're all coding for the same amino acid glycine. Okay, next we come to wobble. So definitely make sure that you know what wobble is for the exam. So we have 61 different messenger RNA codons that code for amino acids, but we only have about 45 transfer RNAs. So the numbers don't match up. So what that means is that some of these transfer RNAs are actually binding to more than one messenger RNA codon. Okay, so let's just take a look at how this works. So here we've got, over on the right, we've got a transfer RNA, and on the transfer RNA we have the anticodon. So we've got CCG, and then down here on the bottom, this is our messenger RNA codon. And you can see that they're complementary to each other. We've got the C's and G's that are matching up. Okay, so that's all right. So GGC, that messenger RNA codon, if you look back on the genetic code table, that codes for this amino acid glycine. Okay, now if we look at this other messenger RNA codon, we've got GGU. And then if we look at our transfer RNA anticodon, we're actually going to use the exact same transfer RNA. So we still got CCG here, and it's also carrying glycine. And if you look at the pairings here, notice that the third base pair here is not following the rules. So you've got a G and a U that are matching up, and that's not supposed to happen. So what wobble is just saying is that with the transfer RNA and the messenger RNA, when they bind together, that third base pair, they don't have to follow the rules. So they only have to follow the rules for the first 2. So that is what wobble is just saying, that they don't have to follow the rules on that third base pair. And that's why you can have some of these transfer RNAs binding to more than one messenger RNA codon. So this one transfer RNA, this same one, is binding to both of these messenger RNA codons. So they're different messenger RNA codons, but the same transfer RNA is binding to both of them. And if you look on the genetic code table, you can see that GGU, GGC, and then also GGA, GGG, they all code for the amino acid glycine, because it doesn't matter what that third letter is. As long as you have the 2 G's, it's always going to code for glycine, because you're always going to have the same transfer RNA binding to it. Okay, so that's wobble. All right, so with translation, we're going to read the messenger RNA codons. We're going to translate or decode them into the amino acids, and we're going to join the amino acids together using peptide bonds to make our protein or polypeptide chain. And translation involves transfer RNA, which is going to bring the amino acid in to the ribosome. And translation is going to occur in both prokaryotes and eukaryotes in the cytoplasm. So I definitely would know where translation is occurring in prokaryotes and eukaryotes. So everybody is going to do this in the cytoplasm because that's where the ribosomes are located. Okay, so this is looking at prokaryotes, and we've got the DNA here. And we're going to be transcribing it with the RNA polymerase, that enzyme that does transcription. And you can see that the newly made messenger RNA is coming off the end of the RNA polymerase as it's going down the DNA. And then as soon as the messenger RNA is coming off the RNA polymerase, the ribosomes are hopping on, and they're going to be translating the messenger RNA even before you're done transcribing. So with the prokaryotes, they're able to do transcription and translation simultaneously. So can this happen in eukaryotes? Can we do transcription and translation at the same time like this? So no way. So there's two reasons why this can't happen in eukaryotes. So one of them is that transcription and translation happen in different places in the cell, right? So remember, transcription occurs in the nucleus, and then translation occurs in the cytoplasm. So you couldn't have them happening simultaneously like this because there's no ribosomes in the nucleus, so this would not be happening. And then the second reason this would not happen as well is the eukaryotes have to process the messenger RNA before it can be translated. So remember, they've got to put the 5' cap on, put the poly A tail on the 3' end of the messenger RNA, and then do the RNA splicing, cut out the introns. So they've got to do all that in the nucleus before they send the messenger RNA out into the cytoplasm to be translated. And we don't have to do any of that processing with the messenger RNA and the prokaryotes. So as soon as they transcribe their messenger RNA, it's just ready to be translated right away. Okay, and then this just shows the simultaneous transcription and translation happening in prokaryotes. And they're able to do this because one, transcription and translation is happening in the same place in the cytoplasm. And then two, they don't have to do any processing of their messenger RNA. So as soon as the messenger RNA comes off the RNA polymerase, the ribosomes can go ahead and just hop on and start translating and making the protein. And we cannot do simultaneous transcription and translation at the same time in eukaryotic cells because transcription is happening in the nucleus and then translation is happening in the cytoplasm, so they're happening in two different places in the cell. And then the second reason we can't do it at the same time either is because we have to process the messenger RNA in the nucleus before we send it out to the cytoplasm to be translated. So we've got to add the cap and the tail on and do the RNA splicing before it's ready to be translated. Okay, let's take a look at the transfer RNA that's used in translation. So we've got the amino acid attached to one end, and it's kind of folded up on itself, kind of looks like a little clover leaf. And we've got the anticodon here on the transfer RNA, and that transfer RNA anticodon is going to be complementary to the messenger RNA codon. So you can see the G's and C's, A's and U's are going to match up. So to fill in our blank here, the transfer RNA anticodon is going to be complementary to the messenger RNA codon. Okay, and then ribosomes, remember, are made up of two things. They're made up of proteins and the ribosomal or rRNA. And remember, the prokaryotes have the 70S ribosomes and eukaryotes have 80S ribosomes, so they have different size ribosomes. Although the eukaryotic cells with the mitochondria, and if they're photosynthetic and have chloroplasts, those organelles used to be bacteria, and so they actually do have 70S ribosomes in those organelles. And the reason that the ribosome size is important is because we do have a class of antibiotics that will bind to the 70S ribosomes and inhibit protein synthesis. And do you remember which antibiotics will bind to the bacterial ribosomes? So remember, those were the aminoglycosides, and then we had the tetracyclines, macrolides, and cosamides, and also chloramphenicol. So just kind of tying the antibiotics in with this chapter. Okay, initiating translation, getting it started. We need to start at the 5' end of the messenger RNA because we're always going to be translating the messenger RNA in the 5' to 3' direction. And the ribosome, the subunits are apart when we start. They're in little pieces. So we're going to have the small ribosomal subunit bind to the messenger RNA at the 5' end around the start codon AUG. And then the first transfer RNA that matches up with the AUG will bind, bringing in the methionine. And then the large ribosomal subunit will fit over that first transfer RNA, and then we're ready to start translating. Okay, so how does the ribosome know which end is the 5' end? Well, it's pretty easy in the eukaryotes because there's that 5' cap on the 5' end of the messenger RNA. But remember that the bacteria don't have a 5' cap on their messenger RNA because they don't do any of the processing. So in their messenger RNA, they've got a special sequence at the 5' end. It's called the Shine-Dalgarno sequence. So the Shine-Dalgarno sequence is located before the start codon here, and that's what the ribosome is going to look for and say, oh yeah, that's the 5' end. That's how I know that I'm at the right end. Okay, that's what they look for in the bacteria. Okay, and it's just named after the scientist that discovered it. Okay, and then there's 3 binding sites for the transfer RNAs in the ribosome, and they're abbreviated APE, and they're used in this order. So I definitely would know this order for the quiz and the exam. So if you just remember APE, that makes it easy. So I would also know what is binding at the A, P, and E sites. So the A site is where the next transfer RNA bringing in the next amino acid is coming in to the ribosome. And then the P site is where the transfer RNA that is holding the growing polypeptide chain is bound. And then the E site is the exit site. So that's where the empty transfer RNA is going to exit the ribosome. So answering our question up here, what catalyzes the peptide bond formation between the amino acids during translation? And the part of the ribosome that does this is the ribosomal RNA or rRNA, because remember it acts as an enzyme, it's a ribozyme, and so it's that rRNA that does that. Okay, so we've got our 3 binding sites in our ribosome. We've got the A, P, and E site. And you can see that the A site over here has the transfer RNA that has brought in the next amino acid. And then the P site has the transfer RNA holding the growing polypeptide chain. And then the E site, that's for exit, that's where the empty transfer RNA is going to exit the ribosome. Okay, so here's how translation is going to work. And we're going to translate the messenger RNA in the 5 to 3' direction. So I definitely would remember that direction. That's an easy test question. So if we take a look on the top here, we've got our A site is empty. So our transfer RNA bringing in our next amino acid is going to enter the ribosome through the A site. And then our transfer RNA holding the growing polypeptide chain is in the P site. And now we're going to make a peptide bond to join the growing polypeptide chain to this next amino acid. And what catalyzes the formation of that peptide bond? So hopefully you said it was the rRNA, the ribosomal RNA. That's part of the ribosome. So that'll catalyze the peptide bond formation. Okay, and then we're going to have translocation, which just means that the ribosome is going to move down 1 codon on the messenger RNA. And now the A site is empty, and our transfer RNA holding the growing polypeptide chain is in the P site, and our E site, our exit site, is where the empty transfer RNA is going to exit the ribosome. And then we just start over, and we just keep going until we get to the stop codon, which you don't need to memorize any of these, but there's 3 different stop codons. And when we get to the stop codon, a release factor comes in, which is just a protein that will make everything fall apart. And the protein gets released, messenger RNA gets released, and the ribosome falls apart. All right, let's put down the differences between the bacteria and eukaryotes and translation. Where it occurs in both bacteria and eukaryotes is in the cytoplasm, and that's because that's where the ribosomes are located. The ribosome size in bacteria is 70S, and in eukaryotes it's 80S. The ribosome recognition site on the messenger RNA, they're looking for that 5' end. So bacteria, they don't have a 5' cap, so the ribosome will look for the Shine-Dalgarno sequence. And that tells the ribosome that that's the 5' end. And eukaryotes, a little bit easier, they have a 5' cap that the ribosome looks for. The start codon, remember, is AUG, and in eukaryotes, the start codon AUG codes for the amino acid methionine. And in bacteria, there's just another little chemical group that's added on to the methionine. So it's just called N-formyl methionine, but it's basically methionine. And then, can transcription and translation happen simultaneously at the same time in bacteria? Yes, it can. And it cannot in eukaryotes, and that's because transcription and translation happen in different places in the cell. Transcription's in the nucleus, translation is in the cytoplasm, so they can't happen at the same time. And then also you have to process the messenger RNA in the nucleus, do the cap, the poly A tail, and do the RNA splicing before it gets sent out to the cytoplasm to do translation.